Anyway, yeah, thanks for the introduction and for the invitation. I'm Josiah. I'm a postdoc at the Kaptein Lab at Utrecht University in the Netherlands. And I want to talk to you today about a project where we combine smart microscopy indeed with optogenetics to try and achieve control of cell biology. And I'm going to start with just what do I mean by control? Because I mean, this is a biocontrol seminar, and we see a lot in this seminar of a lot of different systems biology approaches where we're kind of optimizing feedback loops or gene expression, regulation of proteins. But what I particularly want to do is take a bit of a step outwards because I'm an experimental biologist. And what I want to do is be able to look at a cell down a microscope and try and direct that cell to do what I want it to do. So it's a bit more of this like RC car or robotics type control that I want to talk about today. And I mean, imagine if we could achieve that level of control. So I mean, there are many possibilities in terms of being able to bring cells to the same initial conditions or look at cell-to-cell variation and heterogeneity. We want to be able to derive different biological parameters by learning from this control. We could control cell decisions or different checkpoints in the cell and direct different communication pathways and a lot of more future possibilities. But there are a lot of challenges when you try and do this kind of control, and particularly in mammalian cells. So the mammalian cells have a lot of differences in morphology or gene expression, protein levels at different time points. They can vary in phases of the cell cycle. They have different organelle distributions or different states of differentiation and so on. And it's really this, what I want to talk about today is how we can overcome these different challenges and try and extract some of these exciting possibilities. And the outline of the talk is really I will focus some time on the background and the motivation and talk about how we tend to manipulate cell biology, and particularly with optogenetics, how we can then implement smart microscopy and how people have already, such as Jean-Baptiste, applied closed loop control to really gain this active control of biology with optogenetics. And then I will talk about what we have done with outcome-driven microscopy and how we use outcome-driven microscopy to control directed cell migration and also nucleocytoplasmic transport as two very different applications of this technique. And then I will finish with, of course, some outlook. So starting with some background. So if I, as an experimental biologist, want to investigate the behavior of some cells, what you would typically do is manipulate the protein expression, right? So you can look at from the DNA, the mRNA to the protein level, and then you do some manipulation and you take a fixed cell snapshot or you try and relate this to some live cell dynamics to learn something about the system that you're interested in. And if you want to add some temporal control, then you can do stuff like inducible expression or degradation. You can flow in some drug and look at what happens. Or you can even do inducible interaction, right? So you can add chemically induced dimerization systems to then allow you to look at the interaction between proteins and the effect that this has. But the real king of spatial temporal control is doing light-induced interaction. And this is the field of optogenetics. So optogenetics has really brought us from doing just observation only experiments on the microscope to really actively controlling it. And optogenetics is where you combine genetic engineering with optics to selectively control biological processes using light. And what we do with optogenetics is we take a protein of interest, we fuse it to a light sensitive domain, and we introduce that into our cells. And where do we get this light sensitive domain from? Well, I mean, light sensitive behavior is super common in nature, right? So you think about like phototaxis in algae, like where Euglena have this negative phototaxis, these big Volvox algae have positive phototaxis, and these contain proteins that are respondent to light. And of course, this is also seen in plants with phototropism, where they will grow towards the light to optimize photosynthesis. But how does this work at the molecular level? Well, these proteins typically have a light-induced conformational changes. So it can be that they contain chromophores that are excited by light, and this will lead to a change in state of the protein itself. So this can be, for example, with photoisomerization, where a protein will switch between a cis and trans conformation, or it has a photochemical reaction where you form or break a covalent bond in order to expose different domains of the protein. And through all of these modules that you can get from nature, we've built a whole wealth of optogenetic switches, right? So we're really now able to do control using optogenetics really across the whole spectrum. And it's given us a lot of variation in the types of experiments that we can do and the types of ways in which we can manipulate proteins using light. So for example, at the single protein level, you could introduce allosteric regulation, or you could have a protocleavable protein, or at the oligomerization and dimerization level, you can induce oligomerization or dissociate a complex or dimerize proteins together that you're interested in and assess the effects thereof. And the field of optogenetics kind of started with neuronal biology or neurons. And in classical optogenetics, what you do is you make use of the fact that neurons communicate by action potentials, and that these potentials are set by the permeability of the membrane to ions. So if you introduce light-sensitive ion channels, then you're able to generate or inhibit action potentials to then be able to excite or dampen the communication between neurons. But since then, we've really moved on to, well, not moved on, but in parallel, we're also now looking at how you can use optogenetics to do intracellular spatial temporal control. So now we're really looking at the subcellular level, where you can do, for example, illuminating small regions of your cell to control the orientation of a spindle, or you can recruit a cargo to a molecular motor to induce directed movement of organelles, or you can recruit katana into a microtubule to then selectively disassemble the microtubules in your experiment. And in our lab, we do a lot of these types of experiments, right, where we combine optogenetic modules and we look at subcellular behavior. But what we found was that as we try and get more and more complex in our experimental design, then we found that we need a lot of either prior knowledge of the system. So you need to know that at time point T, I want to illuminate in this specific region with this specific intensity, and you need to know that beforehand. So you need to know that your cell is going to be here in order to illuminate it here, for example. Or it requires some human monitoring, right? So you have to sit there at the microscope and you have to monitor your experiment such that you, after some time, you click in a certain region so that you illuminate that particular area. And so for some time, we were okay with that, and we just got master students to do this human monitoring. But after a while, we looked at how we could introduce closed loop control. So how could we really replace this kind of prior knowledge or human monitoring with microscope-based control? And of course, this brings us to the field of smart microscopy, where with smart microscopy, we perform closed loop control of imaging conditions. And historically, it's used where you perform your analysis immediately on the microscope. The results from this analysis of the image is used to adjust your acquisition parameters. And this will happen in a feedback loop where you first acquire your image, you feed this image into an analysis pipeline, and this pipeline will then work out how to adjust your microscope on the fly in order to optimize your imaging conditions. And people use smart microscopy to do, for example, optimizing your settings of your microscope, such as PSF shaping or adaptive optics. You could detect specific objects, like scanning a large field of view and trying to find an object of interest. You could do this in time as well by detecting specific events, like waiting for a cell to divide and then switching your imaging modality. Or you could adjust illumination parameters like your light sheet parameters over time. And people have used this to great extent. And I think a couple of examples that really show the smart microscopy control across scales are firstly adaptive light sheet microscopy. So here you see an experiment where we're tracking a growing embryo, and we can not only correct for the growth and the drift of the actual embryo, but also correct for different optical properties depending on where you are in this very large sample. And at the other end of the scale, we can look at individual mitochondria and then try and detect their event. So here they're looking at mitochondrial division and then imaging with a slow temporal resolution. But then when a division event is detected, and you're switching modality to image with a very fast imaging speed. And this lets you image these rare events with high temporal resolution without sacrificing sample health or even just the amount of data that you acquire from constantly imaging with fast temporal resolution. And of course, smart microscopy and closed loop, or smart microscopy without closed loop control, has already entered the field of optogenetics. And I tend to classify them, at least in my head, as either passive or active control. And what I mean by this is with passive control, we're looking at feedback that is only optimizing the optogenetic parameters themselves. So you're not looking at the effect of your stimulation on your sample, you're just optimizing where you illuminate or with what intensity. Whereas with active control and optogenetics, you're looking at feedback of the biological system itself. So you're looking at how your stimulation is affecting your biology, and your feedback loop is really aimed to optimize this biological response. And so a couple of examples to show this like passive approach to closed loop control and optogenetics is, for example, in this paper from Jason Town, where they want to continually illuminate on the left side of the cell. So regardless of where the cell is migrating, they want to continually make sure that they're illuminating always on the left. So at every time point, they threshold out the cell, and then they work out the direction, and then they illuminate on the left side. But they don't look at whether your illumination actually affects anything in your sample. And another example is in here where we're looking at, or not we, where they're looking at focal adhesions. And what you do is you segment the cell, you detect focal adhesions, and you want to selectively stimulate specific focal adhesions. Whereas a more active approach would be where you're taking feedback on the biological systems themselves. So one great example of this is in cybergenetics, coming out of Mustafa Kamash lab at ETH Zurich, where you're really looking at then the feedback on the system itself by looking at gene regulation and control of expression by having an internal feedback loop that detects the outcome of your optogenetic stimulus and then adjusts the illumination over time in order to induce specific expression levels. And of course, one great example of this is from Jean-Baptiste, where you can take individual bacteria, you can immobilize them in a mother machine, you can then selectively illuminate individual bacteria according to a regime of how you want their expression to be, and you can then treat each individual bacteria as a pixel in a movie such that you can then represent these individual movies. And all of this prior art has really nicely shown the possibilities of combining closed loop control and optogenetics. But what kind of is missing for us in order to be able to make use of these kinds of approaches for our very diverse set of experiments? So what we thought was what's missing is we want a more generalized platform. We want it to be a bit more compatible for mammalian cells and to overcome a lot of the heterogeneity in there. We are focusing on subcellular dynamics, so really at inside the cells. And we wanted to be able to do not just the intensity of the light, but also really control the spatial positioning itself alongside the intensity. So that was the question for us is, can we build such a diverse smart microscopy platform to really gain subcellular control of cell biology across different scales? And because we want to use this platform to really look at a cell and make it do what we want it to do and achieve a specific outcome, then we termed this outcome-driven microscopy. And the requirements for such a platform were that we want to build a smart microscopy platform, so not just for the kind of outcome-driven approach, but also that can be used for general smart microscopy experiments. We wanted it to be super modular, so we want to be able to pick and change different modules such that we can do a real nice, diverse set of experiments. We had the bold aim to be system agnostic, so that anybody could use this kind of platform and run it on their microscope and their system that they are particularly interested in. I'm an experimental biologist, so for me, I need it to be easy to use and such that anybody can take it and then adjust it for their particular experiment. And it also has to be nice and robust, and it has to work a lot of the time because, I mean, when we're working with mammalian cells, we don't have the luxury of being able to do many thousands of cells at once. We have to really be sure that it's nice and robust and that we can really control our cells with high precision. So a few of the modules that we decided to implement were, of course, a user interface. This is obviously for user interaction. So we've got a very basic UI where a user can have dynamic inputs where they can, for example, select which cell they want to segment and things like this. We needed an image analysis module. So this would be to process the acquired image from the camera and run image analysis, such as cell segmentation in whatever method you want to use for. We need a controller. So something that can process this extracted information and then convert it into something actionable. And of course, we need this microscope bridge, so how we can send commands from our platform to the microscope. And for example, this can be Micromanager, but we've also set up links with commercial microscopes, like we can now control ZenBlue controlled microscopes, for example. And so all of these user adjustable modules are very nice, but they're all quite separated in their own little islands, and you need something to really bring them together and coordinate the experiment that you want to do. So we have this driver module called an outcome-driven strategy, which is where you have an experiment-specific module, and this coordinates communication between all of these other ones. So for example, to link the UI to the image analysis, you need to know when the user has clicked on a cell, which cell is sent to this image analysis for segmentation, for example. Or to link these two, you need to know what information is really is taken from the analysis and input to the controller in order to get it to do its thing. The controller will output a control strength, so we need to be able to convert this control strength into a specific command. So this could be the intensity of the light, or it could be where you illuminate with your optogenetic parameters, or something else like the frequency of the light, or for example. And so all of these are our requirements and our idea about how such a platform could work. And so we set about to do this with great efforts from a very talented master student, Jakob. But it wasn't really until Alfredo came along that the platform was really built into the system that we use today. So Alfredo is a postdoc in our lab. He's also part of NL bioimaging, and particularly with smart microscopy. And he also maintains smartmicroscopy.org. So if you're interested in learning a bit more about the general smart microscopy, then feel free to head there. And what we kind of built was this platform where you can see it maintains the philosophy of the previous modules, where the user can interact with the user interface and also set how they want all these modules to be set up. And then you have this outcome-driven strategy to coordinate the image analysis, the controller, the microscope bridge. And then this Python-based architecture is then able to talk to your microscope through the bridge, such that your platform is able to adjust the optogenetic stimulus. So again, this can be like the power or the shape or the frequency or the wavelength of light that you're using for your stimulus. But we also wrote it such that it can control the hardware of your microscope itself for more general smart microscopy applications, so such as moving the stage around for tracking, or switching objectives or filters for other kinds of event detection type methods. And of course, we need to close the loop. So we acquire our image, and then we send this back into the interface in order to then achieve this closed loop control. And in order to demonstrate this, we had to look at what kind of experiments we could do to really show this outcome-driven control. And so the requirements there were to really try and get some different scales, to look at both spatial and intensity-driven control to demonstrate flexibility. It had to be able to scale up, so you could parallelize experiments and do multiple at once, if needed. And we wanted to use existing optogenetic tools so that people could see what kind of experiments they could do, and show the flexibility of these modules in that you're able to pick and choose them as the experiment depends. So we ended up on demonstrating this with two different examples. The first is with directed cell migration. So in these experiments, we have a construct, I will explain it later, where you illuminate in your cell with blue light. Your cell will polarize towards this direction and initiate migration in that direction. And our goal or our outcome was to really control the migration path of the cell. So be able to draw a path in paint and then make the cell follow it. And our other example is with nucleocytoplasmic transport. So here we have a construct where in the dark state it's held in the nucleus, and when you illuminate with blue light, it exits the nucleus and goes into the cytosol. And our outcome with these kinds of experiments was to control actually not just the cytosolic intensity, but either the cytosol or the nucleus intensity in order to achieve this kind of concentration-dependent control. So I will first talk about the outcome-driven control of cell migration. And just to quickly dive into the optogenetics here, is that we use recruitment of a REC1 effector, TIAM, to the plasma membrane. And what we do is we introduce into our cells two constructs. And the first is this cytosolic construct, which is TIAM1, the effector. linked to a fluorophore and this SSBB domain. And then we also express another fluorescent protein bound to eyelid and then anchored to the plasma membrane using a CAX motif. And what happens is that when you illuminate with blue light, this little alpha helix on eyelid will unfold, and SSBB is able to bind to an SSRA motif inside this eyelid protein. And this will recruit TIM1 to your plasma membrane. And because TIM1 is an effector of RAC, and RAC is involved in actin polymerization and polarization of the cell, this will induce migration towards the region that you have illuminated. So how this looks in our goal and in our feedback loop is that if we want to control migration to a specific path, then we can acquire our image. We can send this to image processing where we segment the cell. We find the position of the cell relative to our ideal path set up as a series of checkpoints. And our control algorithm would then look at where the cell is in relation to this path and decide where to illuminate. And then our intervention is that our microscope will then illuminate that side of the cell in order to bring it towards the path. And because, of course, this is all done in a loop at every single time point, then over time, you can imagine that you can bring your cell along your path just by continually updating which checkpoint you're setting. So the modules that we needed to build were, for our image analysis module, we used segment anything. So this is the pre-trained AI model. We use segment anything and then with a custom implementation for tracking, such that at every time point we segment the cell, there's a little bounding box, and then we update this over time in order to maintain segmentation during our time-lapse imaging. And this was in collaboration with Vincent, who is a PhD student with EOR Small at UU, focusing on AI-based segmentation or general AI-based image analysis methods. And our controller in this case was actually super simple. It's called a direction orientation controller, but all it really is that we have our migration path as a series of checkpoints, and we calculate from segmentation the centroid of the cell. We draw a line between the centroid and our next checkpoint. We make a little cone, and then we only illuminate in the cell this particular region in order to push it towards this checkpoint. And then over time, we adjust where our checkpoint goal is in order to keep it on the migration path. So let's see if it works. So here I am guiding a HD1080 cell around a circular path. So you can see each checkpoint here. You can see in blue the area of illumination and in purple the segmentation. And I will also track the centroid of the cell. And you can see that we very nicely are able to guide our cell around these specific paths. If we also plot the angle of the cell in relation to the starting point, you can see that we are really consistent in our level of control. At the single cell level, if we instead of just plotting the raw angle, if we plot the sine of the angle, you can see we fit a very nice sine wave to show that our control is nice and robust in time. And if we then do this experiment with multiple cells and average the data, you can see that we can really nicely bring many cells along our path with minimal deviation. So we're typically able to keep cells within 5 microns of the path at all times. So what can we use this for, and what kind of how can we push this a bit further? So first, we can look at how cells migrate in crowded environments just to see the effect of other cells and cell-to-cell interaction. So here I'm guiding this cell around at a little circle path, and you see that as it migrates around, it interacts with other cells. They're quite sticky and they pull out bits of membrane, and you can really use this to investigate how cell-to-cell interaction affects cell migration, particularly in the directed cell migration phase. You see here that sometimes the cell is not so happy with it, and then it ended up being dragged away by another cell. It also can lead to some interesting movies. To push this a bit further, we can do it in an even more crowded environment. So here we want to see like how well we can control a cell when it's really being buffeted around by a whole monolayer of cells. So here you see that actually it's very hard to maintain control of this cell during migration because it's really forming adhesions with other cells. It's very hard to drag it away, and it really helps us we want to try and use this to learn something about how cells will do directed migration in a more crowded environment, for example. So if we want to add a bit more advanced control, we can add a bit more of another temporal regulation where what we do in these experiments is I guide a cell around a circle and then I turn off the stimulation, and then I want to know which direction the cell takes, right? Because there are many different hypotheses you could make. So either it could continue off on a tangent to because this is the most recent illumination, or where your most recent or most active GTPase signaling is occurring, or it could continue along the path because it's been depositing some ECM or different extracellular vesicles. Maybe it goes backwards because this is where the most fresh ECM and extracellular vesicles are that it's been depositing. Or maybe one hallmark of cell polarization is the nuclear Golgi axis. So this line between the nucleus and the Golgi is often an indicator of the intrinsic polarization of the cell. So what we did is we did our control, and we guide our cell around a circle. We also label the Golgi, and then we turn off the stimulation and we see what happens. And what we found is that it seems that more often or not, the cell will follow the nuclear Golgi axis, or the intrinsic polarization of the cell. And this is very interesting because, I mean, we've had hours of affecting the signaling of the cell. We've been forcing actin polymerization in a particular direction, but yet as soon as we stop control, the cell is free to move in whichever direction it wants to go, which really can help us learn something about the decision-making of cell migration. Another way that we can show advanced control is by scaling it up. So we could control multiple cells at once. So here you see 3 cells controlled to these little circle paths. And you can see that we can very nicely parallelize these experiments by controlling 3 cells at once, and we're able to keep them all very consistently on their own specific paths. But one thing you might notice is that we got very lucky here where our cells don't run into each other. But so in order to avoid that, we built in this collision avoidance system where we gave each cell a little searchlight. And then if it decided that if it realized that it was about to come into contact with another cell, then we flipped the orientation of control in order to pull it away from this imminent collision. So let's see how this works. So here you see 3 cells being guided around these 3 circle paths, and then you see a little exclamation mark appear when it's detected that it's about to crash into another cell. And then you see the change of polarization and change of migration variably dragging cells away from each other to make sure that they don't crash. So this is a bit more of a fun experiment just to show how much control we have over this system. And we can then use this to learn something about the behavior of individual cells in more crowded environments. So one thing that I've been showing is all of these experiments have been with a constant LED illumination power, whereas optogenetics can often tend to be quite dose responsive. So if you increase or decrease your light intensity, you often tend to get different responses from your cell. So what we decided to do is see whether we could modulate the cell speed by increasing or decreasing the intensity of the light over time. So what we did is we did experiments where we guide a cell around a circle with one particular intensity of light, then we drop the intensity and we see whether it moves faster or slower over time. And then we were able to calculate these averages. And then if we plot this by plotting the change in irradiance on the X and then the change in the speed on the Y, we're able to see that we could induce a modest increase or decrease in speed by just modulating the level of irradiance. So if we illuminate with a higher LED power, then we're able to make the cell speed up. Whereas if we decrease it, then we're able to make the cell slow down. And this is interesting because we can use this to look at kind of limitations in the cells, right? Of whether there's a minimum or a maximum speed that cells are able to achieve, and whether this varies between different cells, and this kind of variation in the heterogeneity between these cells. So one further way that we are using this for a bit more advanced control is looking at 3D environments. So here we've embedded HT1080 cells into a 3D collagen matrix, and then we've attempted to control them in this collagen matrix. And here you can see in this preliminary experiment that we can just very nicely change the migration of the cell along this little channel. And we're interested in doing this not just from the migration and how it behaves through the collagen matrix, and also how it decides on the directions that it should go and how it navigates through this complex environment, but also on how it forms like pseudopods and different extensions of the cell in order to navigate through this environment. So in addition to all of the kind of previous directions that we're working on, I'm also interested in expanding this to more different cell types and systems. So we have some collaborations set up for controlling T cells. So in particular, interested in like controlling T cells to tumors, and then looking at the navigation of T cells around these types of cancer tumor organoids. I'm also interested in collective cell migration. So this is a very cool paper here where they show by transfecting cells with opto they can migrate in an amoeboid-like manner, where even though this is a big group of cells, they just by illumination on the back of the cell, the back of this cluster, you're able to migrate this entire cluster in one direction, which I think is super interesting. We're also interested in looking at how we can use this to do bulk cell control. So now I'm thinking of like inducing gradients where you can manipulate the slopes of the gradient, for example, and the directionality in order to control multiple cells at once without doing the selective illumination and control of the single cells. Okay, so the second example I want to show you is how we use outcome-driven control to look at protein concentration. And here we want to control nucleocytoplasmic transport. And we use this light-inducible nuclear export system called LEXI. And in this case, we have a single construct this time instead of two. And this construct is an M-cherry fluorophore attached to an NLS, so a nuclear localization sequence. And it also has an islet domain attached. And in the dark state, this is held in the nucleus because of the NLS. But when you illuminate with blue light, this alpha helix again unfolds, but this time this alpha helix has an NES encoded. So an NES is a nuclear export sequence. And as we know that a nuclear export sequence is stronger than a localization sequence, the construct is shuttled outside of the nucleus and into the cytosol. And this is nicely reversible. And so we thought that we could use this to maintain intensity at specific set points in our cell. So our feedback loop would look like this, where we acquire our image, we segment out the nucleus and the cytosol, and we calculate the intensity in both. We feed this into a controller that then decides on how much light to illuminate to reach a certain set point. So again, we need to design our modules. So for our image analysis, we again turn to segment anything. But here we encountered a bit of an annoying problem, which is that when you're transitioning between enrichment in the nucleus and enrichment in the cytosol, you end up having this perfectly homogeneous cell where it's impossible to tell where the nucleus is. So you see, like in white, this is what our segmentation was doing, because there's no way to tell which is nucleus and which is not. So what Vincent did is he added in this temporal filter, which then just pauses the segmentation in these cases where it's impossible to segment the nucleus in relation to the cytosol. And this let us have nice segmentation regardless of which state the cell is in. For our controller module, we again have quite a basic controller in a PID controller. And so in this case, our error is just the error of the set point to the ratio or just the intensity that is your goal. And then the control strength, which is the output of the controller, is just directly proportional to your LED power. So such that if you have an increase in LED power, then you would export more. And if you reduce your LED power, then you would end up importing. And you can use this to modulate the levels of import and export over time. But what we found, particularly in collaboration with Menno from TU Delft, is that the basic PID controller wasn't really good enough. So through some modeling and system identification and validation on real experiments, we came up with this model where we have a four-state model where the construct is either in the nucleus or in the cytosol. It's either inactive or active, and we can look at the rates between all of these states. And through modeling and validation, Menno was able to come up with a gain scheduled PID controller but then adjust the gains as a function of the actual set point, such that you're able to you're able to minimize your error at every set point in relation to just the standard PID controller. So how this practically looks is that if we set up an experiment where we try and guide the cytosol intensity to these specific set points, so how we typically represent it is as a relative concentration from the minimum to the maximum that the cytosol can achieve, then we're able to modulate it to like half-fold enriched, 20% and 80%, etc. And in blue, you can see the irradiance as decided by the PID controller, or the gain scheduled PID controller. And you can see that when you need to bump up your intensity, then you just bump up the LED power. And again, if we control many cells and look at the averages, we could really control the cytosol intensity using global illumination with quite high precision. So we're able to guide cells to these various different set points with really minimal error, except of course at these transition points. But we didn't only want to control the cytosolic intensity, we also were interested in controlling the nuclear intensity. So we again made a controller for nuclear intensity and then ran this. And we were also interested to see on how robust it is in the same cell. So running the controller multiple times to see whether there's a lot of variation between control, especially considering that cells have a lot of variation over time and that this might affect the degree of control. But we very nicely were able to show that we can modulate the nuclear intensity to these set points, and that the irradiance as decided by the controller is very similar in each of these runs, but it does compensate in areas when it's needed to be different. So because we built a cytosol and a nucleus controller, then we thought we would try it in the same cell, but then with inverted set points to see what happened. And then indeed we can control the same cell with these two different controllers with 3 runs each, and we're able to guide the cell to these specific set points regardless of the type of controller that you use. And if you average this out, then you get very nice demonstration that depending on the optimization that you want, either on the cytosol or the nucleus, that the controllers are able to then bring it to those different set points. And because they're inverted controllers, then the output from the controller is actually the same, where you need to increase your illumination in order to either increase the cytosol concentration or decrease the nuclear value. So how we can bring this a little bit further is we're looking at targeting specific cells by using local illumination. So all the previous examples were with global illumination, whereas if we look at a cell that's really in a monolayer, then it's very hard to segment. But can we control the nuclear intensity just by locally illuminating that specific cell? And then, yeah, of course, we can. We just, by locally illuminating this nucleus, then we're able to set the relative concentration to our desired set points. So how can we actually use this for something useful? Well, we're interested in controlling protein concentration in vivo as one nice experiment. So we're looking at how we can we have a very nice project in collaboration with Joao, also from Utrecht University, looking at spatial temporal regulation of proliferation differentiation decisions. So here what we did is we introduced the Lexi construct into C. elegans, in particular in the M lineage. And what Joao is interested in doing is modulating the location of transcription factors during differentiation of the worm and being able to titrate them inside and outside of the nucleus to look at how this would affect proliferation differentiation. So in addition to all the previous stuff I've shown on controlling nucleocytoplasmic transport, we're also looking at how we can get more biological insight by looking at concentration-dependent factors, such as transcription factors, and also how we can use this kind of control to derive different rate parameters from our controllers. We're also looking at how we can do quantitative control. So Alfredo, in collaboration with a master's student, is looking at how we can introduce FCS such that we don't just do control over the intensity, but we actually bring this to a more concentration level. Okay, so a couple of slides just on the outlook. So we think that outcome-driven microscopy, or just general closed-loop control of cell biology, can really open up the opportunity for a lot of different experiments. So for example, you can achieve consistency by getting repeated experiments. You can bring cells to the same initial conditions and then let them free and see what happens from there. You can assess the heterogeneity and the causes of deviation from your control and use that to derive intrinsic parameters by doing modeling. And you can then also do robust parallelization of these experiments because you're able to control these cells with high precision. And what I've shown is that outcome-driven microscopy, you can robustly control these biological processes. We can bring cells to defined outcomes, and that we are able to overcome some of this heterogeneity and stochasticity that we see between cells. And as well as all the previous future directions that I've talked about, some of the more longer term things that we're interested in doing are looking at further biological processes. In particularly, it would be very cool to do some multiplex control. So here I'm looking at optogenetic modules that are maybe complementary in wavelengths, where you could, for example, do control of cell migration in one wavelength, and simultaneously in another wavelength, you could induce, for example, secretion. So then you can perform experiments where you're able to guide cells around and also deposit ECM or deposit extracellular vesicles during migration, and then look at how other cells would interact with this kind of path. Our lab is very interested in how we can use optogenetics in more 4D and in vivo type settings. So we're looking at how we can do optogenetics in, for example, organoids. So we've set up some organoid systems where we can introduce optogenetic modules and look at how we can perturb stuff and gain control over those kind of systems. I talked a bit about the segmentation where we use segment anything, but there are some cases where we might need more custom trained models for the segmentation and for the image analysis. And I think one cool direction is also looking at AI-based control systems, so where we really go from the more classical type of control to a more machine learning type scenario. So everybody that's on this slide has contributed in no small part to the data that I've shown today. But in particular, a shout out to Alfredo, who is from NL Bioimaging, and check out smartmicroscopy.org. Jakob, who was the master student that really started this. Vincent, who does a lot of the image analysis, and definitely Menno, who does a lot of the control theory aspects of this project. And if you want to learn more about, in general, the project, or if you want some more details on the control and the system identification, then check out the supplementary in the preprint, and you can read it there or get in touch. That's it. So thanks for listening. I think I'm on time, and let me know if you have any questions.